Hello everyone, how are you all doing? My apologizes on that I stopped the last video without an outro, yeah. But here in this video, welcome back, and we will be explaining the second half of this topic. Beside the diseases, we're not gonna cover the diseases. We're gonna cover the thing we haven't covered. So basically, we're going to cover the Bohr effect and the cardiac cycle. Okay, so let's start with the cardiac cycle. The cardiac cycle is basically the movement of blood through the heart and is carefully controlled by the contracting and relaxing of heart muscles. The cardiac cycle has three stages as follows: the atrial systole, the ventricle systole, and the cardiac diastole. Here, let's cover the definitions for them so it can be easy on us. The atrial systole is the phase in the cardiac cycle following the diastole, where the atria contract and force the blood into the ventricles. The ventricular systole is the phase in the cardiac cycle following atrial systole, where the ventricles contract and force the blood out of the heart and around the body. The cardiac diastole is the phase in the cardiac cycle following the ventricular systole, where the atria and ventricles are both relaxed and blood flows into the atria. So basically, in more details, the atrial systole, where the atria contract, and this forces the atrioventricle valves open and blood flows out of the atria and into the ventricles. Pressure in the atria is greater than the ventricles, so blood is forced out. In the ventricular systole, the ventricles then contract, causing the atrioventricle valves to close and semilunar valves to open, thus allowing blood to leave the left ventricle through the aorta and right ventricle through the pulmonary artery. Lastly, the cardiac diastole, where the atria and the ventricles relax. Elastic recoil of the heart lowers the pressure inside the heart chambers, and blood is drawn from the arteries and veins, thus causing semilunar valves in the aorta and pulmonary arteries to close, preventing black flow of blood. This is the cardiac cycle. But do you know the Bohr effect? Do you know what the Bohr effect is? Before we go to the Bohr effect, let's just revise the cardiac cycle. The cardiac cycle. Okay, great. Let's now go to study the Bohr effect. We have two ways to study the Bohr effect. Either to study the Bohr effect and then study what is transport of gases in the blood, or do the thing vice versa. Well, let's start first with the definition as we did in the last time. Here. Basically, what is the Bohr effect? It is here. A decrease in the affinity of hemoglobin for oxygen in areas with a high carbon dioxide concentration. So here in the graph below is called a oxygen dissociation curve. The curve is sigmoid, which is called as shaped. Why? Because it represents cooperative binding of oxygen. The HB has 4 heme groups to bind 4 O2 molecules. Binding of the first O2 is difficult, binding of the other becomes easier and cooperative due to a conformational change. As HB becomes more saturated, less oxygen can bind, so the curve flattens out. That's what is meant by the partial pressure, the pressure exerted by one type of gas in a mixture of gases. So here the Bohr effect is an effect on HB's affinity for oxygen in differing levels of carbon dioxide. It's shifting of the oxygen dissociation curve to the right. What is the importance or significance of the Bohr effect? Respiring tissues produce high levels of carbon dioxide as they have low levels of oxygen as it's being respired. The high carbon dioxide levels lower the pH, which affects the HB's protein structure. So the HB shows low affinity for oxygen, which means that in those high carbon dioxide areas, the respiring tissue areas, the HB can unload oxygen more readily at the same partial pressure. So basically, here. The Bohr effect is the affinity of hemoglobin for oxygen, which is also affected by the partial pressure of carbon dioxide. Carbon dioxide is released by respiring cells which require oxygen for the process to occur. Therefore, in the presence of carbon dioxide, the affinity of hemoglobin for oxygen decreases, thus causing it to be released. You want to know more about it from the very beginning? Let's go. Hemoglobin. Hemoglobin is a water-soluble globular protein found in red blood cells which consist of 2 beta polypeptide chains, 2 alpha polypeptide chains, and 4 heme groups. Each of the 4 polypeptide chains is bound to a heme group, Fe2 plus ion, to which one oxygen molecule can bind. This means each molecule of hemoglobin can carry 4 oxygen molecules. The oxygen binds with hemoglobin to form oxyhemoglobin and can unbind when needed in respiring cells and tissues. Now, what is the transport of oxygen and carbon dioxide? The affinity of oxygen for hemoglobin, or how easily oxygen loads into hemoglobin, varies depending on the partial pressure of oxygen, which is a measure of oxygen concentration. I just want to tell you that affinity is the tendency of one substance to bind with another substance. Affinity is the tendency of one substance to bind with another substance. So the affinity of oxygen to bind oxygen for hemoglobin, how easily loads onto hemoglobin, varies depending on the partial pressure of oxygen, which is a measure of oxygen concentration. The greater the concentration of dissolved oxygen in cells, the greater the partial pressure. Therefore, as partial pressure increases, the affinity of hemoglobin for oxygen increases, which means that oxygen binds to hemoglobin more readily. This occurs in the lungs in the process known as loading. So where is loading? It's not here. But this is the process. So basically, let me explain it to you. Loading is the process where oxygen binds to hemoglobin in the lungs. During respiration, oxygen is used up, therefore the partial pressure decreases, decreasing the affinity of oxygen for hemoglobin. As a result of that, oxygen is released from hemoglobin in respiring tissues where it's needed. This is known as unloading. As oxygen diffuses into respiring tissues for respiration, carbon dioxide diffuses out and into the capillaries. Here, in the low partial pressure of oxygen environment, carbon dioxide binds to hemoglobin to form carboxyhemoglobin. The deoxygenated blood returns to the lungs where carbon dioxide unloads from hemoglobin, which bind to oxygen again. Do not worry, we do not, we repeat 3 times or something in this video, but if you want more repetition, you can either rewatch the video, or you can wait, or not wait, you can look for the videos where I do study with me, where I repeat a lot, solve many important questions right, and etc. It's gonna be a really good video, inshallah. Like, study actually with me, okay? Here we're just explaining. So now let's complete with the dissociation curves. Dissociation curves illustrate the change in hemoglobin saturation as partial pressure changes. The saturation of hemoglobin is affected by its affinity for oxygen. Therefore, in the case where partial pressure is high, hemoglobin has high affinity for oxygen and is therefore highly saturated, and vice versa. Now, we have factors resulting in different affinities. First of all, the saturation. Second of all, the fatal hemoglobin. But we're not going to cover it from here. We're going to cover it from here. The fatal hemoglobin. Basically, oxygen dissociation curve for fetus is shifted to the left of adult one. Fetal HB has higher affinity for oxygen at all partial pressures because oxygen needs to diffuse from maternal blood to fetal blood across the placenta when their blood flows in opposite directions. Fetal HB binds to oxygen at the same partial pressure maternal HB loses oxygen. Oxygen is needed for fast growth and metabolism of developing fetus. The blood flowing in opposite directions ensures steep concentration gradients to maximize oxygen transfer. This is important as lungs still developing in fetus and fetal tissues have low oxygen levels due to high metabolic demand. The blood clotting process is not going to be covered now. It's going to be covered in the disease part. The vessels were already covered. This was already covered as well. The cardiac cycle here. So, before we come revise from here, let's just cover the things from here. So, basically, the factors resulting in different affinities, saturation, and fetal hemoglobin, as we've said. Saturation can also have an effect on affinity, as after binding to the first oxygen molecule, the affinity of hemoglobin for oxygen increases due to change in shapes, thus making it easier for the oxygen molecules to bind. The fetal hemoglobin, the hemoglobin present in fetuses, has a different affinity for oxygen compared to adult hemoglobin, as it needs to be better at absorbing oxygen, because by the time oxygen reaches the placenta, the oxygen saturation of the blood has decreased. Therefore, the fetal hemoglobin must have a higher affinity for oxygen in order for the fetus to survive at lower pressure. Now let's just finish this part with the Bohr effect, which is the affinity of hemoglobin for oxygen, which is also affected by the partial pressure of carbon dioxide. As we mentioned, carbon dioxide is released by respiring cells, which require oxygen for the process to occur. Therefore, in the presence of carbon dioxide, the affinity of hemoglobin for oxygen decreases, thus causing it to be released. Let's revise. Last time, cardiac cycle. Cardiac cycle is the cycle of repeating events that take place behind every heartbeat and to create every heartbeat. In humans, one heartbeat is equal to one cardiac cycle, which lasts 0.8 seconds. The sound lub dub happens during every heartbeat. Lub occurs when blood is pushed against the atrioventricular valves during the ventricle contraction, and the dub occurs when the backflow of blood hits the semilunar valves during ventricular relaxation. Try listening to your heartbeat. It's gonna be like blub, 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 right? So the cardiac cycle has 3 stages. It starts with the atrial systole, then the ventricular systole, and ends with the diastole, as we've said, which is the cardiac diastole. Systole means contraction, and diastole means relaxation. So when we go with the atrial systole, it means the atria contract, and when we go in the ventricles contract. Lastly, diastole, both relax. So the atrial systole, atria contract and ventricle relax. Blood pressure higher in atria than ventricle. So AV valves open and FV, basically the atrioventricle and the semilunar valves open. Blood flow from atria into ventricles. Atria relax and ventricle contract. Blood high pressure in ventricles and atria. AV valves close and CV open. Blood flows from ventricle into aorta and pulmonary artery. Lastly, the cardiac diastole. Both atria and ventricles relax, AV valves open and SV valves close. Blood flows to fill atria and ventricles, ready for the next cardiac cycle. Now the heart rate. Heart rate is equal to 60 over length of one cardiac cycle. During exercise, the length of cardiac cycle becomes shorter, so heart rate becomes higher. For example, we said one cardiac cycle takes 0.8, so we say that 60 over 0.8. But when it's in exercising, becomes shorter, so 0. Any number. This is not scientific. Let's say 0.6, 60 over 0.6. Okay, so it becomes higher. Ventricles also contract more forcefully during exercise to pump blood at higher pressure. There is a delay of 0.01 second between atrial systole and ventricle systole, because the atrioventricular valves have to close before the ventricles can contract or systole, and to prevent backflow of blood into the atria. Now those we are currently done, but before we actually just say bye-bye, let's make sure. Okay, we did not say the fetal hemoglobin. The fetal hemoglobin is the higher affinity form of hemoglobin found in the blood of a developing fetus. And circulation is that we did it. Oxygen affinity is the continuous relationship between hemoglobin oxygen saturation and oxygen tension. Oxygen dissociation curve, a graph that describes the relationship between the partial pressure of oxygen and the percentage saturation of hemoglobin in the blood. We've covered all that. The venule, a smaller type of blood vessel that connects veins with capillaries. Okay. We are done for this video, and the next video, inshallah, we will be doing the diseases. These questions are gonna be studied together in a different video. Do not worry. Just for the next video, we're doing cardiovascular diseases. Nice to meet you all, guys. Have a nice day. And by the end of the cardiovascular diseases, we're gonna celebrate that we are done with half of unit 1 now, topic 1 all, because topic 1 is biological molecules, the transport, and the cardiovascular diseases. So and by the next video, or the video, we're gonna finish cardiovascular disease, and we're gonna be finished with the topic 1, but we're gonna have still the topic 2 for unit 1 to be over. Have a nice day.